All right, hello everyone, and welcome to the first, let's say, video for this course. So, I want basically what I want to do in a simple manner to kind of help you guys with, studying and getting ready for your exams or either your finals or just for your own leisure, of course, is just to go over the presentations in a more easier fashion and, just make it just kind of help you for or just for your general interest as well. So just a reminder that this is not obligatory. You don't need to listen to these videos. Nothing that I say extra here that you don't find in the presentation will be in the exam, so no worries. Really, if I say extra examples or extra stuff, that is purely for interest sake, nothing else. Okay? So let's start off with the first chapter of this course, which is the general structure and classification of viruses. So we just want to go talk, in a very general manner. What are viruses? How do we classify them? And their kind of structure and what do they contain and everything like that. Because that's going to help us later on to go into more specifics into every single family of virus by knowing just the general structures. Because for me, LB is so much easier to say like, oh yeah, this is an enveloped virus. You already know it's like, oh, enveloped. It means it has a lipoprotein outside, then it has also the capsid. And if I say it's like, double-stranded what's it called, DNA or something, you already know what that kind of means. So this is very important to kind of build your foundation, in my opinion. So let's go into really the history to the concept of a virus. You know, the first person really to say what a virus was Edward Jenner in 1798, and he was the one who basically introduced this term, virus into microbiology. And what virus technically means in Greek, literally, it means poison. So this is the story and how he actually came up with that kind of name. So Edward Jenner was, let's say, a very wealthy person back in the 1700s, and basically, he had a very big house. He had a huge kind of farm or plantation, and we had a lot of milkmaids. And this was during the time where a lot of people were dying from smallpox, a very nasty virus that infects humans that used to really cause a lot of deaths. But he realized that these, milkmaids were actually somewhere they kind of immune against smallpox. And they started to realize that they got infected with a different, very similar virus, which is the cowpox. And this cowpox, similar to the smallpox, it gives you these kind of small little kind of dots all over your body, similar to the smallpox. But cowpox is quite it's not that lethal. It's very harmless for humans per se. So he noticed like, okay, but why are they? So what's this thing? So he wanted to actually test this. So what he did, which I have to say is very unethical. Nowadays, this would never happen. He literally took one of the kids of these milkmaids, and he literally took a bit of the fluid from a vesicle that you find in these basically from these milkmaids who had cowpox. So they had these basically vesicle fluid, these massive vesicles which were like filled with fluids, and inside that fluid there's a lot of virus in there. So what he did is he literally took a bit of that and he injected it into the boy. Then he took this boy and he literally threw the boy into a ward or basically a room in a hospital that had a lot of cases of smallpox. So this is super unethical because you're throwing a kid into a room that he could potentially die. Let's imagine that he was not actually, you know, immune against the virus. That boy would have died. So nowadays that would never have happened. But anyway, he did see that the boy developed sustained immunity against smallpox, so he did see that yes, it worked. And then he basically labeled whatever that was inside that kind of liquid in the vesicle as poison, virus, because he kind of had this in his head, like, okay, I most probably added a virus that was so similar to the virus that we were getting infected, and that's what's giving this immunity. And that was the first form of a vaccine. So technically, Edward Jenner, other than just being a person that brought virology into this whole microbiology world, he also kind of gave the first invention of the first vaccine ever. Because viruses per se can be vaccines. They can be natural vaccines, so they can also, you can genetically modify them to create the vaccine. I do this quite regularly. That's basically most of my job back in Finland to create uh, viruses as vaccine platforms for cancer. And Edward Jenner was the first person that actually, said the first person to say to do that. So that's where, the concept of virus first ever came. So small general characteristics of viruses. Viruses are in the range of 20 to 300 nanometers. 20 nanometers is the biggest protein you have, and 300 nanometers is the smallest cell we have. So this is just to give you a range of what a virus is. Is as, bigger than a protein, I would say, because of course the smallest virus is about the biggest protein, but it's basically not the biggest virus is as big as the smallest cell that we have. So it's in between a a cell and a protein, the size. Viruses have nucleic acids, they have genetic material, and these either can be DNA or RNA. Never both. It's either one or the other. They do not have hybrids. They do not have both at the same time. No, it's either one or the other. Viruses consist of nucleic acids surrounded by a protein coat. Because these viruses have genetic material, which is DNA and RNA, these are susceptible to nucleases. This could be DNases or RNases, enzymes that cut DNA or RNA respectively. So it's super important that a virus has some sort of structure to protect it. And that's called a protein code that we call actually a capsid, which I'll be talking about much more in details about it. Some viruses have additional envelopes, a lipoprotein envelope that mostly comes from the host cell, which I'll also be talking a bit more extra about it. Viruses do not have cellular organelles like mitochondria or ribosomes. They do not have it. Literally, a virus is lipids. Sometimes it's lipids because if they have, again, this lipoprotein, proteins, and genetic material, nucleic acids, either DNA or RNA. That's it. They do not have any kind of, let's say, ordered complexity. They do not have proteins that come in together to create the mitochondria, the powerhouse of ATP. No, that doesn't exist. Okay? Viruses, hence, are obligate cellular parasites. They replicate only inside cells. They are obligated to become parasites, meaning they need basically some other living form to live off from. That's what a parasite means. They are hence cellular parasites. They need live cells to actually live. And one important thing to always keep in mind is that if a virus infects that cell, it doesn't do anything. It cannot replicate. It needs that cell to be alive. Keep that in mind as well. Viruses replicate through replication of the nucleic acid and synthesis of the viral protein. So I want to go through these three and give you examples here. So viruses do not multiply in chemically defined media, and viruses do not undergo binary fission. Viruses, as I said, are obligate cellular parasites. They do not or cannot uh, duplicate or grow just in basically a media filled with food. Bacteria can do that, but viruses cannot. They need to actually infect a cell or a host organism to actually create, uh, to multiply. They do not go under. They do not go on binary fission. Binary fission is literally the process and where you have the cell membranes basically splitting apart to create two. That bacteria do that a lot. Viruses do not do that. They cannot. How they replicate is again by multiplying the amount of nucleic acid and viral proteins. That's how they do it. They get inside the cell, they hijack the host cell's DNA machinery, RNA machinery, or protein machinery to create multiple copies, and which will they come assemble and create new viruses. So these are very important characteristics to know about viruses that I need you guys to know. So let's talk about the genomic material of viruses. So as we said, they either are RNA or DNA. So they have nucleic acids. Again, either RNA or DNA, they're never both. These nucleic acids can be linear or they can be also circular. DNA, if a virus has DNA, it's always a single molecule. Okay? So it's just one molecule, either be it linear or circular. If the virus has RNA, it can be either a single molecule or in several pieces, multiple small pieces. And a very, very good example of that is influenza. Influenza actually has several pieces of RNA. For some reason, I think it's nine. Do not, I could be wrong, but if not mistaken, I think it's nine pieces of RNA or seven, something like that. But this gives it an advantage. Think about this actually. If these kind of, why do viruses have pieces while others have single molecules? But for influenza, it's actually quite useful because if it has these small pieces of RNA, it's so much easier to kind of, let's say, kind of lose one piece and gain another piece from another virus. So influenza, there's multiple different types. And of course, these pieces of RNA are different between type 1 and type 2, let's imagine. So imagine you have type 1 virus coming in and type 2 influenza virus coming in. They can exchange small pieces of RNA so quickly. To exchange a whole molecule or the whole genome is extremely hard. So that's why it's quite hard for the viruses to exchange information. So what happens is that you can actually, this influenza viruses can exchange small pieces with other influenza viruses, and it can actually gain new functions. And I give you here a very good example. There's a lot of viruses, there are a lot of influenza viruses, the human viruses, that basically will infect the basically humans. And they infect the humans, and they only go for the upper respiratory tract because they bind to certain kind of sugars that have a certain configuration, which is I think 2,6. That's usually the configuration they go for humans. In the ones that basically can go to birds, they go to a different configuration, which is 2,3. That one can go into the lower respiratory part of humans. So what happens is that pigs can actually get these two different viruses. They can do a recombination of them, and suddenly you have a new virus that's able to bind to both. And this actually can go inside into a human and actually start replicating in the upper and also go down to the lower respiratory virus, the lower part, and that can cause pneumonia, which is extremely dangerous. Very good for the virus, but extremely dangerous for us. So that's one good, actually, positive thing for RNA viruses that they can do. They can actually, exchange materials and actually evolve into, let's say, better or more lethal versions of themselves. One thing I also want you to know is that almost all viruses contain one single copy of their genome. So they're haploid, meaning they only have one copy. We humans are diploid, we have two copies of a gene. There's one exception of these kind of viruses, that's the retrovirus family, that has two copies, diploid. And these are HIV viruses. And this is again another extra benefit because if you have a copy, it means that if you mutate one gene, the other gene can still help and produce the protein. That's extremely important because some viruses are extremely susceptible or sensitive to any mutations. Because some mutations can destroy the whole gene and you only have one copy, so you cannot create any more of that protein. While with the HIV, you have an extra copy that can actually still create a functional copy of that protein. So viruses can be divided into two large groups, so RNA-containing viruses or DNA-containing viruses. But we like to use a more different classification, which is the Baltimore classification, which is basically divided into six. The first one is double-stranded DNA viruses. Two is single-stranded DNA viruses. Third one is double-stranded RNA viruses. The fourth is single-stranded RNA viruses with a positive strand, so positive polarity, 5' to 3'. Single RNA viruses with a negative strand, hence negative polarity, so the opposite, 3' to 5'. Or six, single-strand RNA viruses associated with the enzyme reverse transcriptase. So I'll give you examples of each. So you have the double-stranded DNA families, which is extremely important. You have the poxviridae, the herpes viridae, hepadna, adeno, and papoviridae. So I'll give you examples of each one. So the pox viruses basically are quite known, to create these kind of viruses that are basically they're called molluscum contagiosum. These you can get them actually from dirty pools. They're harmless, but they have these they look like warts, but they're not really warts. Molluscum is an example of that. The herpes viridae family is the herpes simplex viruses, which we all basically have either seen people have them. By the I think the age of 40, 80% of people have herpes viruses or even not, if not everyone. It's only 20% of people that have herpes viruses that actually get these cold sores. Actually, so people don't know why. It's a very interesting kind of case. But that's an example of the herpes viridae families, the herpes viruses. This is most probably herpes simplex type 1, because usually that goes for the mouth, but it can also be type 2. Type 2 also can go into the mouth, but it's mostly found in your genitals. Then you have the hepatnaviridae, which are basically your hepatitis viruses, like the hepatitis C. That's an example of it. Then you have your adenoviridae, which is your adenoviruses, and these are extremely important in just getting colds. They are viruses that you can get a cold from. And the papoviridae viruses. These are the most important one from that family is your HPV, so the human papilloma viruses. And these are viruses that are sexually transmitted, and they create these kind of litchy warts that you see over here. Extremely annoying, very not they're not painful, but they're just very annoying, very ugly looking, and you can pass them basically through sexual transmission or through direct contact. They're extremely infectious. So they're viruses, the HPV viruses that create warts, they don't usually cause cancer. But if you there are different types of these papilloma viruses that do not cause warts, but they can cause cancer. Specifically in women, they are the sole reason why women have cervical cancer. So this is why now there is the new vaccine against these papilloma viruses, because are able to almost eradicate cervical cancer. But it also can cause other cancers, for example, like anal cancer. It could also create oral cancer, throat cancers, and also in some cases, penis cancer for men. Not as common as cervical cancer, though. Then you have basically your single-stranded DNA families or your double-stranded RNA families. Your single-stranded DNA family is your parvoviridae family. These are extremely important. You get them mostly in dogs. They have the parvoviruses, which is extremely lethal for these dogs. And then you have the double-stranded RNA family, which is the Reoviridae family, is most important from them, which these reoviruses also causes a lot of these kind of different respiratory infections. Then you have the single RNA families with a positive strand. These are cool viruses because the single strand of RNA can directly be used as an mRNA because it's already in the positive sense. It can be directly, used as, already a transcript to be translated into the protein. These include the picornaviridae, calciviridae, astro, corona, flavi, toga viruses. Most important one, as it says here, basically, is the coronavirus, the coronaviruses, the ones that we basically threw into the pandemics, the SARS-1, SARS-2, MERS. These are the ones of the viruses that I'm talking about that have a single-strand RNA with a positive strand. Then you have your single-strand RNA families with a negative strand. These are basically your orthomyxoviridae, paramyxo, rhabdoviridae, and filoviridae. The viral genome does not act as an mRNA because remember, it's, the negative sense, the negative strand. So it has to be transcribed back into mRNA. So all of these viruses contain an extra protein, which is a viral enzyme, transcriptase, which then is able to take this negative strand and turn it into an mRNA. So a RNA that is in the positive strand. So they, again, like I said, they all contain this enzyme transcriptase. An example for this is basically from the orthomyxoviridae family is the influenza viruses. And these influenza viruses have a single strand RNA with a negative strand. And like I said before, these influenza viruses have pieces of their virus, of their genome. It's not one full piece. It's divided into pieces, into smaller pieces. And the final one is single-stranded RNA viruses that are associated with the enzyme reverse transcriptase. These are ones that have a single-strand RNA, but they have an extra enzyme that is called transcriptase that is able to take this RNA and transcribe it into its complementary DNA. It's able to reverse transcribe it. Big example is the retroviruses, and a huge example of those are HIV. HIV does this quite well. So now let's talk about the structure of these viruses. So at one point when people were looking at, were able to look at viruses and look at the structures, they were calling them all different types. They were calling them bullets. They were calling them basically cubes. They were calling them bricks, etc. But honestly, we started to look at more their structure, and they have this very particular structure that we basically started to put them into different categories. So before I get into the specifics of this, I want to go into some terminology to help you understand, what I'm saying. First terminology that I'll be using is virion. Virion is the whole complete virus particle. So it's the whole thing. It's basically the capsid, it's the basically this envelope, it's the DNA inside, everything. It's the whole package of the virus. Then you have a capsid. As I said at the beginning, is that viruses have DNA or RNA, so nucleic acid. And these nucleic acids need to be surrounded by a protein coat. That protein coat is called a capsid. The addition of the protein coat and the nucleic acid is called the nucleocapsid. Okay? So nucleic acid and the capsid, put them together, literally fuse together nucleic acid and capsid, you get nucleocapsid. And then you have the capsimers. Capsimers are the structural protein units that make up the capsid. So the capsid, as we said, are literally made of proteins. Well, it's made of capsimers, literally a unit of protein that is repeated to create the capsid. I'll go into more details about this, don't worry. So let's look into the capsid. As we said, viruses have the nucleic acids, DNA or RNA, and they need to be surrounded by a coat, a protein coat that's called a capsid. So helps it protect it against other enzymes like DNases or RNases, but also other physical conditions that are on the outside. This capsid is made of structural units called capsimers. These capsimers can either be one protein or multiple proteins, but they form one capsimer. If let's imagine they are made of multiple proteins, they all come together and they form one capsimer. So one unit of a capsimer, and this capsimer is repeated to create the capsid. So as you can see here, all of this kind of, let's say, balls over here, that's one capsimer, and they're basically all repeated and stuck together to give structure to the capsid. Same thing goes here. The capsid here, these are these kind of triangles that come together to create, the whole capsid. So why are capsimers identical? As I said, capsimers, they're literally repeating and they come together to create the capsid. But why are they identical? Why is that? Something about nature that is so beautiful is that it has evolved for good reasons. That there sometimes it is like that because there's a good reason that why it was like that. And in the case of capsimers, why they are identical is that it reduces the need for extra genetic material. So now a virus doesn't need to encode for many different genes to create different proteins to create the whole capsimer, the whole capsid. No, it can actually just encode for one protein or just a couple of them, 3 or 4, to create a capsimer. And that's it. So it doesn't need to have a huge DNA or RNA or nucleic acids in its genome to encode for them. So basically, viruses are small. They cannot actually take in too much DNA. There's a certain limit. So by making the capsids identical, you limit the amount of DNA can then fit into a small, tiny little virus. Second, it promotes self-assembly, meaning that these capsids, because they're identical, they will stick to each other. You do not need enzymes to come in to actually put them together. You don't even need energy. Just put them together. People have done that. People have taken actually nucleic acids and purified these capsimers and put them together in a test tube. That test tube had no cells, no enzymes, no energy, and literally they were able to create a virus because these capsimers were able to self-assemble. So they got together, they surrounded a piece of DNA, they got together and were able to create the capsid because the capsimers are able to self-assemble. And that's a very good thing because, again, these viruses cannot create energy. They need a source they they need a source of energy. They cannot create it, or they cannot basically they just cannot afford it. So having this basically capsomers able to self-assemble is quite efficient for them, or like energy efficient. This is just an example of an unenveloped basically viruses, the adenoviruses. There's a very beautiful capsid that it has. Bit blurry image, so I'm sorry for that. But some viruses just have capsomers. I mean, a capsid. That's it. And usually we call them naked viruses or unenveloped viruses. So if you hear me ever say unenveloped viruses, it means that they have just a capsid. And if I say also naked viruses, it's the same thing. They call them naked because they don't have an envelope. And then you have viral envelopes. Some viruses have an envelope. This envelope is literally made of virally coded protein and host lipid. Okay? So what happens is that a virus will create its capsid, with the basically with the capsimers all together, and then it's gonna bud off literally from the host's lipid membrane, and it's literally gonna take a piece of the lipid bilayer or the lipid membrane, and that's gonna basically encapsulate the capsimer. So again, as I said, it's literally made of coated protein and host lipid. Most of basically the lipid, the viral. So one thing that I also want to say is that the capsid over here, the capsid and the basically viral envelope are kind of attached together, or let's say kind of interact together through another protein called the matrix protein. So viruses that have a envelope have a matrix protein. They have to. And basically, it's literally this black ring over here that kind of helps the viral envelope kind of interact with the capsid. So always remember that enveloped uh, viruses have to have a matrix protein to kind of connect the viral envelopes with the capsid. And as I said before, the viral envelope is acquired as the virus exits the cell. So it's a process called budding. So once you create basically the capsid, the virus is going to go to this host cell membrane and it's literally going to bud off by and also stealing a bit of that lipid that's going to encapsulate the virus. These are examples of the herpes, the rabies viruses is here. This is the influenza viruses. This is the herpes viruses. This is how they look like. So this is a very beautiful picture of the herpes viruses where you have really kind of this capsid, literally looks like a capsid, and then you have basically this envelope of lipid, and then basically and also protein in there as well. Here again, as I said, there is this matrix protein that helps this interact, the capsids interact with the envelope. So the thing about these viruses is that the envelope viruses is that people might think that by gaining an extra, let's say, kind of envelope or extra membrane would give it a bit stability. Not really. Actually, it confers instability. It gives the virus less instability. So really, non-enveloped viruses are more stable compared to enveloped viruses, much more stable. Because what happens is that now this virus has a lipid membrane. The lipid membrane is much more unstable than protein. That is basically that is formed in the capsid. So the envelope, the lipid membrane is actually very sensitive to heat, to drying, to detergents, and lipid solvents like alcohol, compared to the naked virus. So these viruses actually are the non-enveloped viruses, sorry, are much more resistant to alcohol or to heat or drying compared to enveloped viruses. And this actually has a certain clinical relevance because viruses that transmit via the fecal oral route, so either through the mouth or they're basically through basically the mucosal areas of the anus, it actually these viruses are mostly uh, are naked, meaning that they're non-enveloped, and it's simply because they go through a very harsh environments, very harsh environments actually. So they need to be able to survive, especially a lot of basically drying, a lot of heat actually, and different types of detergents that are being exposed. So they're not able to actually go through that route. So it's usually naked viruses. Mostly the envelope viruses interact or infect through the blood or through direct contact because they're not allowed they cannot really survive outside. I'll give you an example. Adenoviruses. I play a lot with adenoviruses, and adenoviruses are quite resistant. You can literally put them on a table, and they can live actually for the next until the next day. If I take, for example, this the herpes viruses or this lentiviruses, and I put it outside, it's gonna die. It cannot survive that much the heat or basically the dryness, while the adenoviruses can because they do not have this lipid membrane that is so sensitive. So do not get mixed. This envelope kind of gives instability. Extra viral proteins that I want to talk to about is basically, as we know, you have the capsid proteins that protect the genome from degradation of or nucleases. You have also other proteins in the capsid that are basically mediating the attachment of virus on the host cell. And this is extremely important because these proteins that are on the surface of this virus are the ones that are super specific and they bind to specific cells. This determines the species. Either this virus actually infects humans or cats or dogs because these surface proteins that are binding or doing this attachment, they are extremely important because they will bind to specifically, they can only sometimes, for example, bind to just human proteins. So let's imagine they only bind to they call it this CD46 as a receptor, usually found in animals. And a lot of adenoviruses will only bind to human CD46. They will not bind to dog CD46 at all. They won't. So it can determine species, but it can also determine organ specificity. And it does that because CD46 is mostly found in certain organs. It's not found everywhere. So that's why, for example, the herpes viruses has a very good tropism towards the liver they have a perfect protein, the liver cells, that the herpes viruses has a protein that can attach to it. So it can give this a certain specificity. Some viruses have enzymes inside the virion I'll give you an example of some enzymes that have actually that have actually enzymes inside the virion, which is single stranded RNA viruses with negative polarity because they need to have the enzyme transcriptase. Remember this? They need to have enzyme transcriptase. Not all of them, basically not all viruses have, but all single stranded RNA viruses with negative polarity have it. Retroviruses and hepatitis B viruses, they actually need reverse transcriptase because they need to convert their RNA into complementary DNA. So they actually have enzyme reverse transcriptase. Some weird, actually, viruses have something called super antigens. And super antigens, it basically means it's actually the super comes that it's able to activate your immune system extremely high. And specifically, it activates a T cell that's called CD4 T cells, which I'll be talking a lot much more into details a bit later. But these CD4 T cells, they are, you might start thinking, it's like, okay, well, why does a virus want to increase the amount of CD4 T cells? Because some of them need it to replicate. An example of this is, for example, the EBV virus is the Epstein-Barr virus that causes mono, is a nice example of that. The Epstein-Barr viruses need CD4 T cells actually to replicate. And some proteins have something called tegument proteins. Uh, Tegument proteins are found between the envelope and the nucleocapsid, and they are basically kind of factors that regulate transcription or translation, either from the viral or cellular processes. So these are proteins that are literally kind of in some way kind of factors that help transcription translation. So one thing I also want to talk about is serotypes within a virus. Some viruses basically have different serotypes, and now what that serotype means, it's basically a subcategory of a virus based on the surface antigen. So I'll give you an example, okay? So let's imagine here you have polioviruses. You have the blue poliovirus and you have the red poliovirus, okay? These are the same viruses, but they're different serotypes because their surface antigens, so the antigens or the proteins outside, are different. So here you see this is basically a bit more kind of, let's say, open. This has more of a pointy edge, more triangular. This has more of a soft kind of edge. This gives basically a bit of a different, it's the same virus, but it gives you a different serotype because it has a different surface antigen. Some viruses actually have just one serotype, and others actually can have, you know, three or hundreds. Measles, for example, has one serotype. Polio has three. Rhinoviruses have 100. And this actually can be very clinically relevant. And this can cause immune evasion. So I'm going to use here an example of Streptococcus pneumoniae. This is a bacteria, but you literally apply the same concept that I'm talking here to a virus. So here you have Streptococcus pneumoniae, okay? All of these are Streptococcus pneumoniae, but are different serotypes. And you can see outside they have different shapes. For example, the triangle that's in yellow, this has basically a square in blue, circle, basically that's in purple, another triangle that's in basically this kind of light green, and etc. They're all Streptococcus pneumoniae, but they have different surface antigens, hence there are different serotypes. So let's imagine that you get infected with this Streptococcus pneumoniae that has basically this yellow triangle. You create an immune response and you create T cells that are against specifically this surface antigen, or you create an antibody response that is basically binding specifically to this triangle. Your immune responses are usually against surface proteins. They're always against surface proteins. Keep that in mind, okay? So now you created this immune response. You have the antibodies, you have the T cells against specifically this yellow triangle. You get infected with Streptococcus pneumoniae, but a different serotype. So this serotype has a different surface antigen. So these antibodies that you created against the yellow triangle are not able to bind here. And you need to go again. So you don't have any memory. So this is actually very important because it doesn't mean that if you're basically, infected by one Streptococcus pneumoniae, that you're protected by other Streptococcus pneumoniae because they can have different surface antigens, and different surface antigens require different immune responses. So this, in medical implications, this is extremely important. So I'll give you here the example of polio. Polio has basically three, as we were saying. Here, basically, it has basically three serotypes. And what that means is that a person can be immune to poliovirus type 1, but still get the disease, poliomyelitis, caused by poliovirus type 2 and 3. Just because someone has the antibodies of polio type 1, it doesn't mean it's protected against type 2 and 3 because they have different antigens on the surface, and the antibodies created against polio type 1 do not bind to type 2 or 3. 2. The polio vaccine must contain all three serotypes in order to be completely protective. So, if you want to create a vaccine that's able to protect you against all of polio, you need to create one that is basically has all three serotypes because you need an antibody response and a T cell response against all three of them to be fully protected. In the case of measles, you need one. In the case of rhinoviruses, you need more than 100. And this is actually creates some sort of bottleneck because some vaccines are then hard to create because some of them have too many serotypes, like the rhinovirus is 101. That's almost impossible to put them all together in one. Or some of them have they have their surface antigens, they keep changing constantly. So they have so many different types of serotypes that you basically numerically it's hard to keep up with. An example is HIV and hepatitis C virus. These are extremely hard to create a vaccine because their surface antigens keep changing, hence they have so many different types of serotypes. So one thing that I also want to talk is about the symmetry of viruses. So as I said at the beginning, is that people back in the day used to call viruses, bricks, bullets, spheres, etc. But no, viruses can be divided into three groups, and that is depending on the morphology of the nucleocapsid and the arrangement of the capsimers. So how the nucleocapsid looks like and how the capsimers are arranged. Extremely important. The three groups is based on morphology of the nucleocapsid and arrangement of capsimers. Okay? So the first basically group is called cubic symmetry. This is when the virus is icosahedral in shape, meaning it's almost spherical particle, and basically the nucleic acid is contained inside the capsid. So inside the capsid you have the virus, so the nucleic acid. And this icosahedral particle is composed of 20 equilateral triangles, meaning that these triangles are equal all in sides. They have 12 vertices and can be in rotation symmetry 2, 3, or 5, and that's basically how the capsimers are arranged. So either that they have a symmetry of 2, 3, and 5. And what that means is simply is that when you look literally from above the virus, either basically you have two triangles or three or five. That's what it means, fivefold, threefold, or twofold in symmetry. These are cubic symmetry. They literally look like spherical particles, and they are made of 20 equilateral triangles. Then you have more the helical symmetry. These are basically capsimers that are arranged around the nucleic acid. It's not inside, basically it's around it, meaning that, it's basically, they're kind of just going around the nucleic acid. And this gives it a spherical, basically, no, sorry, not spherical, a more elongated or pleomorphic kind of structure, more like kind of a spiral. So this is a pure example of a helical symmetry. So you see this is going like this. The capsimers are going around it. So that's why they have a spherical kind of thing. It's not that basically in this kind of case, this capsid, the cubic symmetry, you have the capsid is formed and the DNA is just inside of it. No, this is going around it. Okay? So that's the helical symmetry. Their capsimers are arranged around the nucleic acid. And then you have the third, which is complex symmetry. These viral particles do not conform to either cubic or helical symmetry. They don't give a crap, literally. They don't care if they're helical or complex. No. A very nice piece of information for you guys to have. If you see a DNA virus that is human, that it only infects human, it's basically helical. All basically DNA viruses that basically bind to human are helical in structure. For some reason, there has to be, I'm quite sure that evolutionary wise, there is a reason for that. That I most probably will look up for it, but I don't know it yet. So these are again the cubic symmetry, look very beautiful, and cube is like literally 20 different equilateral triangles all together. This is basically the adeno. There's the herpes viruses with basically that has, as I said, this really nice envelope. These are the helical symmetry that you see basically with influenza and rabies viruses. They literally look much more kind of helical. And these are more the kind of complex symmetry. This is an example is the pox virus, that they do not look neither helical and neither do they look spherical at all. So this is where I'll be stopping for today.